Imagine a world where the mighty woolly mammoth roams the Arctic once more, and long-lost creatures reclaim their place in nature, or visiting a real-life Jurassic Park. Science is on the brink of resurrecting extinct species, but should we? The promise of reversing extinction dazzles with possibilities, yet the risks and ethical dilemmas loom large. Are we playing saviors, or are we tampering with forces beyond our control?
De-extinction is a process of using biotechnology to revive species that have disappeared. This concept, also known as resurrection biology, became feasible thanks to advancements in cloning, selective breeding, and genetic engineering. One key technique involved is somatic cell nuclear transfer, or SCNT, first proposed by Hans Spemann in 1938. In 1952, Robert King and Thomas Briggs successfully transferred nuclei between frog cells, laying the groundwork for future cloning efforts. The most well-known application of SCNT was the cloning of Dolly the sheep, the first mammal cloned from an adult cell. Scientists accomplished this by extracting the nucleus from a mammary gland cell of a Finn Dorset sheep and inserting it into an empty egg cell of a Scottish black-faced sheep. After 6 days of development in the lab, the embryo was implanted into a surrogate mother, leading to Dolly's historic birth.
This technology helps in many ways. It may offer insights and breakthroughs in biotechnological advancements. Hence, it paves the way for revolutionary medical treatments, enhanced genetic research, and sustainable solutions in agriculture and environmental conservation, which can help us in assimilating new insights on genetics, evolution, and natural resources that are currently not available to us. Being able to clone species from the preserved DNA is a major step forward in genetics and bioengineering. It can help protect endangered species and extinct animals while also giving us valuable insight into Earth's history. With this technology, scientists may predict future events like ice ages, find better ways to care for the planet, and many more. These advancements could be integral for both wildlife conservation and human survival.
Take the woolly mammoth, a creature that lived more than 10,000 years ago. If we were able to bring it back, would it be able to survive in today's world, with pollution, global warming, and habitat loss? So if it can't adapt, then what even is the point of bringing something, a life, into this world for it just to die in pain due to disease or illnesses? According to the National Institution of Health, Dolly the sheep, who was cloned and born in 1996, developed arthritis at a very young age and suffered from ovine pulmonary adenocarcinoma, a type of lung cancer, premature aging, and shortened telomere length. Due to these illnesses, Dolly only lived 6 years, while the normal lifespan of a Finn Dorset sheep is 10 to 12 years.
With the introduction of cloning technology becoming more mainstream, many laws and regulations have been added by the Canadian government regarding the issue. In 1989, new reproductive technologies such as IVF were created, leading the Canadian government to consider the ethical, economic, and social issues with the technology, leading to the Assisted Human Reproduction Act, which was finalized in the law in March of 2004. Some points to consider within the AHR Act are that children's health and well-being must be considered first. Free and informed consent must be given prior to the application of any AHR technologies. People who use AHR technologies must not be discriminated against. Abuse of AHR technologies will be criminal, etc. With many regulations and preventative measures put into place, the Canadian government has done a good part in reducing the amount of misuse of AHR technologies and other related events. Human cloning is 100% illegal in Canada, and if found guilty of committing any crimes related to AHR, it can be dealt with $500,000 fine or 10 years in jail, or both. Although the Act has limited new scientific advancements from being developed, our AHR technologies are still ranked highly in the world, competing with top countries such as the United States of America, China, UK, and others, all aiming towards the same goal of assisting humans for the better.
With a better understanding and base of assisted reproduction in general, We can decide if we do want to revive an extinct species with the current technologies and knowledge we have on hand. Many factors play into choosing which species to clone or not, such as the reason for extinction, if it will benefit us, if it will benefit the environment and local ecosystem, as well as many more.
To summarize, the idea of de-extinction as a new phenomenon promising new areas in biotechnology, medicine, and the environment draws the attention of researchers to its social, ethical, ecological, and practical consequences. The concept of bringing back extinct species like the woolly mammoth sounds fascinating, but we need to carefully consider if these animals will be able to survive, or if we are just cursing them to life that will never be worth living. Other forms of legislation, such as Canada's Assisted Human Reproduction Act, are of utmost importance as they protect against unethical practices and public endangerment that can be brought by developments in cloning and other. By taking these steps, decisions on whether or not to revive an extinct species should be approached with attention to ethical reasoning, life, and scientific questions. The questions that need to be posed do not solely rely on can we, but redirect to should we.